Good afternoon, everybody. Today I'm going to be presenting the research I've been working on for the past year and a half on evaluating Ficus sirica and Psidium guajava as antimicrobial agents. So to start, I want to begin by defining antimicrobial resistance, or AMR. It is essentially the evolution of resistance mechanisms due to selective pressures by bacteria. So let me break that down a little bit. Let's say you're sick, you're prescribed ampicillin. So you have a population of microbes. 5 are randomly sensitive to ampicillin, 1 is randomly resistant. So you take your antibiotic, the sensitive microbes will die, and the resistant one will survive and reproduce, contributing to a population of resistant microbes. So you can see how it would be a problem to be prescribed antibiotics when they're not needed. You needlessly contribute to the evolution of these resistant populations. So a recent study showed that over 30% of antibiotics are prescribed unnecessarily, contributing to this issue. AMR is a growing global threat. Currently, there are around 2.8 million infections each year in the United States alone, and this problem is projected to get much, much worse. This is a graph showing numbers of deaths associated with a particular cause in 2016, and then the projected deaths associated with AMR in the year 2050. And I've highlighted that in red. You can see that there were over 700,000 deaths associated with AMR in the year 2016. This number is projected to rise to over 10 million deaths by the year 2050, and that's deaths annually. I would like to compare this briefly to cancer. Cancer is the leading cause of death worldwide. It kills around 8.2 million people each year. AMR is projected to pass that by the year 2050, maybe sooner. Okay, so people may say AMR is clearly a healthcare problem, but that's not all. It's also an extreme economic burden. According to the CDC, 6 out of the 18 most concerning AMR pathogens cost the US more than $4.6 billion each year. So, the prevalence of resistant infections have increased. This is a graph showing the percent resistant infections over total infections between the years 2000 and 2020 for 2 different pathogens, and I will get into these a little bit more later. You can see that between the years 2000 and 2020, both the prevalence of resistant visa and resistant multi-drug Pseudomonas aeruginosa have increased by quite a bit. So this data is showing that AMR is a huge problem. We need to find new strategies to combat it, and that's where my research comes in. So I look at plant extracts. So plants are biochemical factories. They have a ton of different compounds that have a lot of beneficial functions. So first, I'm going to be talking about Psidium guajava, or guava. Guava has antioxidant activity, meaning it has the ability to prevent the accumulation of or to degrade reactive oxygen species, or ROS. And ROS can cause health issues in and of themselves. Additionally, guava has antimicrobial activity, which is why it was selected for this study, as well as it's important for cholesterol and blood sugar regulation. Guava also has antioxidant and antimicrobial activity, but it also has numerous cardiovascular benefits. So what are my research questions? First, I wanted to know, do these extracts have antimicrobial activity in my hands? And then I wanted to know how cellular morphology, or cellular shape, changes when the cells are treated with these extracts, and use that information to begin to understand the mode of action of my extracts. And I'll get into this a little bit later, but the mode of action is essentially how an antibiotic inhibits bacterial growth. This is an overview of my methods. So first, I needed to create my plant extracts. Then I needed to determine if these extracts had antimicrobial activity. This was done via Kirby-Bauer disk diffusion. Then I quantified that activity via minimum inhibitory concentration and analyzed morphological changes associated with treatment via bacterial cytological profiling. In the future, these results will be analyzed via principal component analysis. So, in order to prepare the extracts, the fig leaves were fresh, so I dried them, ground them into a powder, and then agitated with methanol for 72 hours. after which I rotary evaporated the solution and was left with a sludgy substance. I resuspended this substance in 50% dimethyl sulfoxide to a concentration of 50 mg per ml. The guava already came dried, so that was ground into a powder, agitated with either methanol or ethanol for 72 hours. Then it was rotary evaporated and resuspended in 100 mg per ml DI water. So first, I needed to know if these extracts had any antimicrobial properties at all. This was done by Kirby-Bauer disc diffusion, or KBDD. So essentially, I inoculated an agar plate with a confluent lawn of my bacteria of interest, added dried impregnated discs onto the plate, and then incubated overnight at 37 degrees Celsius, which is body temperature. Then the next day, I measured the zones of inhibition to determine if my extract had any antimicrobial activity. And these are my results. It's kind of a lot, so I'll walk through it step by step. So first, I looked at E. coli LPTD 4213, or E. coli. And E. coli is a gram-negative bacteria, meaning it has a cell membrane, a peptidoglycan layer, and then a lipopolysaccharide layer on top. So it has 2 layers of protection beyond the typical cell membrane. This is a mutant E. coli that has holes poked into the LPS layer, making it so that this bacteria can be effectively treated with a treatment. So all 3 of my extracts inhibited the growth of this E. coli. I then looked at various strains of Pseudomonas aeruginosa, or P. aeruginosa. P. aeruginosa is also gram-negative bacteria, and I found that my guava methanolic extract was effective against all 4 strains of P. aeruginosa, while the ethanolic was effective against 2 of them. I also looked at vancomycin intermediate Staphylococcus aureus, or VISA, and VISA is gram-positive, so it does not have a lipopolysaccharide layer. It only has 1 extra layer of protection. And I saw that both my guava extracts were effective against both VISA strains tested. So a KBDD gives a binary yes or no answer. Does it have antimicrobial activity or not? In order to quantify that, I had to perform a minimum inhibitory concentration, or MIC. So first, I prepared serial dilutions of my extract and inoculated a 96 well plate. Then I added my bacterial suspension, incubated for 36 hours at 30 degrees Celsius, and then added tetrazolium red. Tetrazolium red is really cool because it dyes actively respiring cells pink. So, in other words, if a cell is inhibited, it will be clear, but if it's not inhibited, the well will turn pink. And this is an example of what one of my plates looked like. It has decreasing extract concentration going from left to right, and I've highlighted what I determined to be the MIC in a red box. So you can see where it goes from being clear to pink, that's the MIC. So these are my results in a little bit more detail. Briefly, what even is an MIC? It's the minimum concentration of extract required to inhibit bacterial growth. So a lower MIC means that an extract is better at inhibiting bacterial growth. Looking at these results, we can see that the guava ethanolic extract had an MIC of around 156 against the E. coli, while the fig had an MIC of around 2,500. This indicates that the ethanolic guava extract is more effective at inhibiting the E. coli than the fig. I then looked at PA14, and you can see that the ethanolic guava has an MIC of around 2,500 micrograms per mil, while the methanolic extract has an MIC of around 1,250. This indicates that the guava methanol extract is more effective at inhibiting growth than the ethanol. Finally, I looked at VISA-1, and you can see that the MIC is exactly the same in the methanolic and ethanolic extract, indicating that there is no difference in the ability of the extracts to inhibit bacterial growth against VISA-1. So from there, I was able to begin to determine morphological changes associated with treatment and begin to identify a mode of action. A mode of action is essentially how a treatment inhibits bacterial growth. So first, I grew the E. coli to a late log phase. This is essentially when it's most susceptible to treatment. From there, I treated with 5 times the MIC of my substance of interest. And these are my treatments. I had 5 of them. The first one was untreated, so this is a negative control. And then I looked at 2 known antibiotics, ampicillin, which has a known mode of action of inhibiting cell wall synthesis, and tetracycline, which is a known mode of action of inhibiting protein synthesis. I also looked at fig and guava ethanolic extract. So then, after I treated with 5 times the MIC, I dyed my cells with DAPI to stain the DNA blue and FM464 to stain the cell membrane red. From there, I was able to visualize these cells via confocal microscopy and begin to analyze these morphological changes via principal component analysis. So these are my BCP results. So you can see that the untreated cells are small, their bacilli are rod-shaped, and they have genetic information that fills the majority of the cell. This is expected. Then we have the ampicillin-treated cells. These exhibit an elongated morphology, which is consistent with previous studies. Then, when I imaged the tetracycline-treated cells, they also exhibited an elongated morphology, which is not consistent with previous studies. And they had that condensed genetic information. You can see it really well in the bottom tetracycline image. And it's essentially the idea that the genetic information is not taking up the whole cell. When I treated my cells with guava and fig extract, with both of them, they exhibited an elongated morphology as well as condensed DNA. But these are just images, right? We need to be able to quantify things. This would be done by a principal component analysis, or PCA. It essentially is a way to group things based off how similar they are to each other. So more similar would mean more clustered together on this graph. And I would do that by measuring different morphology characteristics, so DNA length, DNA width, cell area, things like that. And I would be able to graph this data onto a principal component analysis plot like this and be able to determine, okay, fig has a similar morphology change to ciprofloxacin-treated cells, or guava has similar changes to ampicillin-treated cells. So that's my next steps. So in conclusion, the KBDD revealed that all of my extracts were effective against the E. coli, while the guava extracts were effective in varying degrees against the AMR strains from the CDC. And then the MIC results showed that the guava extract was more effective at inhibiting the E. coli than the fig, and there were minimal discrepancies between the guava extracts overall. Additionally, guava extract was better at inhibiting VISA than PA14. And for BCP, the extract-treated cells exhibited elongated morphology as well as condensed DNA, but further quantification is needed in order to identify mode of action. So these are my references, and these are my acknowledgements. Please let me know if you'll have any questions, and also feel free to email me. Thank you all for listening.
Very, that was incredible. I'm going to open up the floor for questions for Mary, and I'm going to sprint as fast as I can through the first question to grab your scroll to realize computer confusion with the store. But don't ask too good of a question without me. Your first question for Mary, this was so
Yes.
Dr. Deere.
So you use leaf extract for both.
Yes.
Do you know are the effective compounds in only the leaves, or they have the fruit too?
They are also in the fruits. I don't know if they're the same compounds, but the fruit themselves do have, yes, antimicrobial compounds.
And then with your profiling miniatures?
Yes.
Some of them, is the DNA, is like that one cell in the DNA is fragmented, or is it just
It-
an artifact of the depth or something?
Yes, it does appear that it's fragmented. Yes. Yes. Yeah.
Especially with a lot.
Yes.
Sorry,
No, go ahead.
Would you expect that fruits of similar composition to your guava or your fig would show similar results?
It's hard to tell without running experiments, you mean like fruits with also antioxidant activity or like,
Yeah, like do similar things or have similar.
oh, I would assume so, but more experiments would need to be done. Yeah. Yes.
So you use ethanol and methanol.
Yes.
Do you have any idea why there's a difference between them?
The only thing I can think of is that the chemical composition of methanol and ethanol are slightly different in that one has, I think it's like an extra carbon or something. So maybe that impacts it a little bit, but I'm not entirely sure.
Yeah, I'm curious, because could you go back to just the results with the methanol versus the ethanol?
Yes.
A little further. The one with the graph. The one with the curving bar.
Yeah.
I'm trying to think, because everything seems like there might be some soluble ethanol but not ethanol. So
Yeah.
the ethanol sometimes works. So there's multiple
Or
compounds, one
it-
of which is not soluble ethanol.
It could also be that the different strains from the CDC have different like properties as to what can get into the membrane,
Yeah.
things like that. But I'm not sure.
And zones of inhibition frequently don't correspond to MICs.
Yeah.
MIC is way more accurate.
Yes.
Can you show on the picture with the MIC and with the color, when you showed the wells and you
Yes.
said this is what you determined to be the cutoff,
Mhm.
do you make that call visually, like you just can't see the red?
Yes.
Okay.
Because the indicator we use, it dyes actively respiring cells pink, and so if there's no color, we can assume that there's no growing cells.
And it's just like by the naked eye, like this is where I can
Yes.
see it.
Yes. We do have a light box that makes it a little easier to see, but yes. Yes.
I have a kind of silly question, but I really liked your graphics for your protocol.
Oh,
How
thank
do
you.
make that?
Oh, it's BioRender. Yes, I love BioRender.
Well, if there aren't any other questions at the moment, I'm going to invite Dr. Bainum, who loses 2 points. It's one, even though that was an amazing presentation. I'm going to invite Dr. Bainum, who worked with Mary on this project, up to say anything she'd like about working with Mary, and also to give her the honors cord and stole that Mary will wear at graduation in recognition not only of this incredible project, but everything she's done as an honor student at St. Edward's. So congratulations, Mary, and thank you, Dr. Bainum.
Thank you. Yeah, so a lot of you probably know Mary well, and you know she's an amazing, outstanding student. It's been an honor to work with her. Anybody that's ever worked with me knows that you have to be very independent to work with me, and you have to really take ownership of the project yourself. It's not that I do it and they watch. She didn't really tell you how difficult this project was. Every one of those pictures were the result of multiple days getting to the point of being able to visualize it. At some universities, they have image centers where somebody runs the microscope and images your samples for you. We don't have that. She used the microscope and she imaged the samples herself. And when she applies to applying to medical school or MD, PhD, people are going to be shocked that as an undergraduate, she was able to use a confocal microscope. It's amazing. She presented her work at the ASM branch meeting recently and won an award for her poster presentation. So she's definitely represented incredibly well. And I look forward to seeing what she's going to do next.